Hey, Robert. Hey, Andreas. It's Allissa. It is July 5th, 2025, and a lot has happened this week. So, the last time we were together, um, we ended with Mandy Harvey explaining how she had a connective tissue disorder, and at 18, she lost her hearing, but yet she can sing. Oh, she can sing beautifully. So, it goes along with the idea that at 4 years old, she was teaching herself how to sing pitch, timbre, rhythm, cadence, all of those things that Nina Kraus talks about in Of Sound Mind, the book. So, um, a lot of, like I said, a lot of things have happened. Um, to date, it's 22 years ago that my dad found a new love of his life, and now I have a bonus mom. They were married on this day 22 years ago. My daughter wasn't even, she was an egg and a sperm, but not even together. They were in separate little areas. So she wasn't she. She wasn't. She wasn't my daughter or a person. Um, I'll get to her later uh, because she and I had a date on Wednesday, and we were in Carl's Corona in the coffee shop, and she explained something to me, and it's just like, oh my. First of all, I don't know how she's gotten so smart. But secondly, she's curious, and like we've talked about, she listens to um, lots of different sources, and then she groups all those sources together, and she comes up with her own way of thinking, and I absolutely find it fascinating. I've learned so much from my daughter. She is the experience of a lifetime because I couldn't learn any of this stuff in school. No way. Okay. Um, she explained something to me about me, and I guess I'm not the only one because I talked to my dad yesterday, and he's like, "Pfft." And my bonus mom was sitting next to him, and she just laughed and smirked, and she's like, "Oh my God, that's your dad." And my daughter also recognizes it. So I'll explain. My daughter calls it linear thinking. I kind of changed it to um, branched linear thinking. And it's this situation where we have an idea, we have a task, and we start on it. And then uh, a little shiny thing catches our eye, and then we go towards that, leaving the task here unfinished. And then this shiny little thing over here in this linear direction, we work on for a little bit, and then something else shiny shows up, or another thought pops in, and we go that way. And so we're leaving all these little crumbs. And this is so me. So if I start something in the morning over here, it might be 10 o'clock at night that I come back to that thing that I started, or 3 days later, because I'm doing all these other things, but they had to get done. And this branched linear thinking is exactly the problem I'm having with finishing these letters in a concise form. So, oh, I don't know if you can see that, but a little message came up. Dr. Sally Sutton's birthday is tomorrow, July 6th. She passed away last year. Last year in June, my mom passed away, June 4th. My aunt passed away, her sister, in July. Um, and then Dr. Sally passed away right after her 96th birthday. 92, 96. That must have been 96. Man, she was a sharp lady. She was an osteopath. So Andreas, she was an osteopath, and she fixed so much food for my mom and I. And she's the one that helped me live with my neck and treated me for close to 3 years so that I can actually survive all my 5 car accidents, and I can now treat my own neck because of her. Boy, did I just digress. Okay, so branched linear thinking is probably not a term that you're ever going to be able to look up and say, oh, that. But it describes the way that this works in here and all these little squirrels running around on treadmills. But if I'm in a linear thought, I can forget to pee, I can forget to eat, I can forget that I have an appointment. I can, yeah, because I've zoned out. I've cut out all this extra noise and I'm in the zone and it feels good. So I'm going to try to get in the zone and finish these letters. This one's called video letter 10, which is the Roman numeral X, and Ardem. That's our goal. So because I'm in branch linear thinking mode, our goal is here, and this is where we're going to end up. But you know what? Life goes on. And yesterday, I was listening to this. Can you see it? I have a really slow computer. It's 12 years old, and I love it, but it needs to find a new home. It's called I can't send this picture anymore to my computer for some reason. The leap, the leap, Mars question mark. It was a miracle we got to Florida. So this is what I listened to yesterday. It's from National Public Radio, Science Friday. I think Science Friday, and these programs might not be here in a year because the National Public Broadcasting in the United States is going to have most likely their funding rescinded. But it's a great story because Steve was the coordinator for the probes that went up to Mars, and he had a huge task. So Steve was a team leader, and as Jennifer said, he took a different approach, and that's what all these videos are about: is taking a different path, taking a different approach. It's not because things before were wrong; it's because we can relearn and do things a bit differently and take a different direction so that medicine can be more efficient, people can feel better, they can learn things about themselves that they didn't know. And I took a class with Julian Baker. It was a 5-day dissection class, and there was this really smart lady there, and she said, we don't know what we don't know. And that's exactly it. My vision, it wasn't until October. It's just like my eyes are getting tired. They're itching a lot. I can't look at the screen anymore. And lo and behold, my eyes are changing. Chronologically, I was born in 1972, so I'm 53. And yeah, the the hyaluronan and the fluid and the glycans and all this other stuff in there, it's altering the shape of my eye. So yeah, I go to an eye doctor and I have an eye test, and they put in a puff of air and they had me read a bunch of letters, and then they gave me a bunch of little squiggles, and lo and behold, I need a new prescription of glasses. So I got these progressives so I can always have them on. But then I had to relearn with progressives. If you're looking down and you're jumping from rock to rock with the kids at the school, you're looking down and looking up, looking down and looking up. And that's kind of weird because with these progressives, the center of my eye is helping me with near, and then the rest of it around is helping is just promoting my far vision because I can see far away without a problem. But without these glasses, I couldn't even see the little camera that's on my screen. See, this is that branch linear thinking. Boy, did I go off in a different direction, but I'm trying to come back. So I got these glasses I've relearned and I've retrained my brain how to handle that looking down and looking up. It took a while, and I did feel a little sick and had headaches, but that didn't last long because I got a pretty smart body. I like it. I want to keep it. Okay, we're going to go back to the leap. Great show. So, Steve, too much light. I got a good idea. Why don't I just go on the computer and show you it? Duh. Okay, hang on. Okay, found it. So join me. So let's see. We have here Science Friday, and there's two ways. You can see the URL, and you can go down and you can click here to listen, and then you'll get a screen and just listen. But then you can click on this that says read story. So then you go to, if you want to read or if you want to listen, it's up to you. But loves Ira Flato. Ah, ah. And this is a good one too. Jurassic World Rebirth. It's all about the movie and how scientists are the stars and the heroes. And thank goodness, finally, scientists are the heroes because they are. We we couldn't have survived this COVID virus and had a vaccine so quickly. Um, well, we could have survived. Nature always finds a way, according to Jurassic Park. Um, but we survived better because we had a vaccine that was found very, very quickly. And it wasn't a politician thing. It was scientists. They found out how to help us cope with that virus. Yeah. Anyway, this Jurassic World birth, the they talked to the paleontologist that was brought on for the movie to make sure the movie was correct. And he talks about how just in the recent few years, they've relearned that the Spinosaurus is not the Spinosaurus that we saw in our dinosaur books when Robert and Andreas when we were young. Instead, it's actually bigger than the T-Rex, and it swam most often in the oceans with this huge long tail that swung side for side, this muscular tail that helped propel it through water, and it ate mostly fish. Pescatarian. Okay, so here we go. We have the leap Mars. It was a miracle we got to Florida. So this is Steve, and his geologist Steve Squires risked his career and millions of dollars to get two rovers roaming on Mars, but the mission didn't make it to the launch pad. Okay, so NASA engineer Jennifer describes many obstacles the team faced with spirit and opportunity. So the reason I wanted to bring that up is because at 11 minutes, when you listen to it, Steve says scientists and engineers had to work together on this team. And he's brilliant when he talks about this. Where's my piece of paper? Where's my piece of paper? Oh yeah, here it is. See, it's awfully messy, but wrote it this morning. My brain just works better in the morning, especially after a little coffee. So the leap, he says, scientists are seekers of truth, and there's no limits. So Robert, you're getting this video letter because you're the scientist that is seeking the truth. And boy, are you a great team leader. You bring groups of people together cohesively and you empower all of us to keep being curious. And then there's the team on the team are engineers. And this was Steve's team he was talking about. So we have scientists and engineers. Engineers are practical inventors and creators. And their creations that they make need must work. So I think I'm not an engineer. Andreas, you're probably closer to an engineer than I am because you're an osteopath. So you're creative. Um, uh, you're not putting together a human, but you're putting together bits and pieces of a puzzle of why their body is doing certain things, and then you're trying to um, invent a way to have their body work cohesively together. So the reason I bring this up is because Steve said something really cool. He said, you put scientists and engineers together and there's tension. Now, when the tension is lagom, Swedish word lagom, just right, then these two different worlds get along great and they can do amazing things. And Robert and Andreas, you've already done amazing things. And Robert, I know why Andreas is your new hero, because he's pretty cool with that research that he did with the low back study and the ultrasound and emotion. And emotional regulation is very important that we learn. So if you have the tension between scientists and engineers just right, that's good. And Steve, being the person that he is, and Jennifer says instead he when the engineer's creation didn't work, he didn't come to the group and say, "Why is this not working?" He he empowered the group. Okay, what can we do? How's it going to work? Let's take a step back, even though they had a deadline. But because they had a deadline, this tension can be, instead of cohesive, this tension can be acidic. And when that tension is acidic, it's not cohesive, it's corrosive. And like our body, if something's corrosive, it's lytic. Lytic is a Latin term for breaking down. And we do want to have things break down because that's the way nature works. Everything breaks down and it builds up, and it breaks down and it builds up. But you want it in balance. So when this tension became acidic and emotions flared, the project didn't get done. So you have to have a team leader come in and like, let's take a step back. So this video letter about Ardem is taking a step back. And now I think we can get to a better place. So let me collect my thoughts, and hopefully I'll remember to push the play button, but I just have to pause a little bit. I remember to push the play. I've done it several times that I haven't pushed the play button and I can't go back. So, so I remember to push play. I'm having my Tazo tea, which is not a product placement, but it's lemongrass and it's helping to calm my nerves. So I will say, based on what I thought with the LEAP podcast from Science Friday about Steve and Jennifer, the NASA engineer, I can say, Robert, you are the scientist. Andreas, you are the engineer. And what I find me being is I'm behind the scenes. I'm the puzzle putter together. And I've used a collection of various different sources to come up with information that I think can benefit both of you in your research and how you address the public. And there is a critical need, and Ardem's going to say this in a little bit. Yeah, and he says it much better. There's a critical need for people to understand or to know what they don't know. And I didn't know my eyes were causing a problem with my tiredness, my itchiness in my eyes, and my headaches. But until I got the glasses and the different prescription, things worked better. So if I didn't have a vision test and I didn't pay for the new glasses, that was October. I, who knows what I would be doing? I wouldn't be looking at the screen, that's for sure. Anyway, so let's go on with Ardem, because the whole point of this is him. The whole point of this is him. He's uh, I've written to him many times. I have, and he's got somebody nice in his office that has answered me because he's a busy guy. Okay, so let's just give a little background. And the background is important because of who this person is. And who this person is, Ardem. Yes, he's a professor at Scripps. Oh my goodness, what a wonderful thing to do. But here's the thing, he's curious. And his early life experiences shape who he is because he grew up in Lebanon and came over the United States at a young age. And when he was in Lebanon, there was a war. Now, as he explains to a Lebanese podcaster, he was young enough to know there was a war, but he was young enough to have the people around him put him in a safe situation. So there was groups of people huddling together to survive the bombings. And what he remembers of that time is everyone being together working cohesively. And they did that so that the children were safe. And if that doesn't give you shivers, then okay, gives me shivers. But because of that early childhood experience, whether it's conscious or unconscious, he is a great team leader. And this scientific discovery about PSEOs could not have happened without his leadership. Well, it probably could have happened, but it happened better because of his leadership. And there's a whole team of people behind him. And as you go along, so he got his lab got the Nobel Prize in 2021, but also David Julius and their lab got the prize. And then David Gitney, American neuroscientist and developmental biologist. Yeah, he also is instrumental in a lot of this research. And then Ellen Lumpkin, and she's an American neuroscientist and professor of cell development and biology and neurobiology. And she's also important in this whole scheme of things. Oh, and you know what? I'm gonna take one of those branches. I know I was in a linear thought, but I'm branching. This is important. I don't know. Hang on. So here's a branch from my linear thought, and we're going to go to my email because emailed to me, is that gonna, nope, 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 inbox, yes, inbox. Um, this one, yeah, earwax secretions may help detect Parkinson's. I don't know if I'm going to get to that, but that was really cool. Okay, so you you too, I don't know what that means, but this is, I think I have the wrong one. Yeah, here it is. 2025 Annual Force-Based Manipulation Investigator Meeting. The Force Net Team, Bridging the Gap. So this is all about force-based mechanisms. So they're coming together, and it's Friday, July 18th, and hopefully I'll see you there because it's free. And if I stop sharing and then I share again, we can look at the draft. So here is the draft information. You don't need to see that. There. I thought Ellen Lumpkin was in here. Oh, Dr. Alex Chesler. You really want to hear what Alex Chesler is talking about, because Alex Chesler has done so many awesome videos on YouTube about NIH, and he's worked with Karsten Boneman. Oh yeah, he's the one that did the thing about the hand. So he talked about how the piezo and cells respond with pressure, and he and his team came up with these these lasso receptors that are on the skin that are connected to our hairs, and soft touch doesn't activate them, but a heavy pull of our hair like this, a yank, activates these lasso uh, receptors. I'm going to screw up the name, so I'm not going to go there, but I have information on him, and maybe in my branches I'll get that up. But here's the thing, these lasso receptors are not only stimulated by hard, heavy pull of the hair, they're also stimulated by blue light. And if that doesn't give you these crazy feelings of, wow, I mean, so we think about that, and we think about, okay, well, our skin uses the sunlight to make vitamin D. Well, these lasso receptors are also stimulated by blue light. So we have another therapy type. It doesn't even have to be pressure of our hands. It can be a phototransduction. So eyes phototransduce. Pressure receptors mechanotransduce. So these mechanoreceptors that they have does both. It phototransduces with blue light, and it mechanotransduces with heavy pull. So incredible. And then there it is. Yep, neurons matter. Dr. Helen Lumpkin. Oh, and there's Greg Gerling. So, yes, they're very important to this whole scheme of piezoreceptors and proprioception. So very glad that I can go on wild goose chases without you having to follow me. But finally, I found information about Dr. Alex Chesler, and where he talks about these receptors that neuron responds to pulling of a single hair. So he says, hi, my name is Alex, and we're focusing on so I thought for sure I had this up. So specialized mechanosensory nociceptors mediate rapid responses to hair pull. And it's in the journal Neuron. So I'll let you do whatever you need to do. This is another a really awesome one that talks about the same thing. So those are people in his lab that discuss this. But this is encodes painful information, and it's a fundamental thing on how we learn and how we feel. Um, but we don't know what we don't know until we have a scientist that finds something out. And who knew that there's a mechanoreceptor that's also sensitive to phototransduction? And that was Dr. Alex Chesler's. So please go to the July 18th force-based manipulations principal investigator, so you can see Dr. Alex Chesler's work. So you can see Ellen Lumpkin and David Gitney's work as well. So is it time to get back to Ardem? Oh my gosh, yes, yes. And in my little plight of trying to find things for you, I also found I also found the oh unheard stories. I think you can see that. Oh, I sure hope you can. But it's the Lebanese podcaster. Unheard Stories presents Nobel laureate Adam Pataputian, Dr. Lubana Hassiana. I'm sorry if I pronounced that wrong. But at 7 minutes and 36 seconds, she says, we treat many diseases and ailments, and we'll be talking about that in a little while. But she talks about Dr. Adam Pataputian's academic footprint, his past life experiences, and this discovery is going to lead to so many different generations. And what this discovery about how proprioception and piezoreceptors give us a sense of the world around us is incredible. So, gosh darn it, let's go back to Ardem's Nobel Prize lecture, because this is necessary. Okay. I don't think I'm sharing that screen. So you have to look for that green line. If you don't have a green line around it, you're not sharing the screen. So we just talked about Dr. Ellen Lumpkin. Um, so definitely Ardem Pataputian does a shout out to David Gitney and Ardem Pataputian and David Julius. Of course he does, because he's a team leader and he knows it's not just him. So we know much less about the sense of touch, which gathers mechanical, thermal, and chemical information from peripheral tissues such as the skin and translates into an electrical signal that neurons understand. More generally, most cells in our bodies communicate through chemicals, but cells also respond to pressure. And how this is accomplished has remained unknown. So this whole lecture is to talk about how it works. And at 2 minutes and 44 seconds, this is incredible. Our perception of reality is defined by our senses. So we've been talking about this, and that's why that blue figure is there. And the blue figure is there because that's how we figure out the world. Time and time again, we're going to go over this. Thank you, Royal Carlson, for making this for me. But this is so important as massage therapists, body workers, but also in people that are studying pain. All of these receptors, when they couple and synchronize together, like Yuri Hassan shows us with metronomes, this makes us work. And we forget, being a small child and all the times that we fell down, but somehow all that falling and those failures taught our body how to maneuver in the world and how to utilize different receptors in different ways to get our muscles to move and coordinate their action so that we can sit and we can balance, so we can gesture like Mr. Roy Carlson has done here. We can cross our legs. We can also chill out. And keep in mind, just like Alex Chesler has found, these mechanosensory receptors, not only can they mechanotransduce, some of them phototransduce with blue light, but others are polymodal. And what I mean by polymodal is that they have different personalities. So when Doug Nelson does a podcast with Whitney Lowe, and they talk about nociception, when nociception is high, doesn't matter what kind of input we're putting in, their personality is saying danger, danger, danger. And it's not in her head. We're not imagining it. It's real. And that's what Laura Mosley talks about. And I like him for that. Mm. But all of this synchronizes, couples together. So then we go to the pyramid of learning. And in this pyramid of learning, here's our central nervous system, but we get the information from the periphery. The periphery is so important to how this works. And the inventors of the pyramid of learning, this is a model. And where are my ladies? Williams and Schellenberger, 1996. 1996, pyramid of learning. So using all of our senses that Ardem just said, we couple them together, we grow, we develop, we put more a collection of various information from different sources, proprioception, vestibular, tactile, olfaction, visual, auditory, and taste. And then we start building up, and our awareness of two sides of the body starts to happen. And we have more motor planning, and we can actually navigate in the world all on our own without somebody teaching us. And we have postural security. And this happens rather fast for a baby because they learn to hold their head up. Not because necessarily, well, the muscles do get stronger, but they have a want, need, and desire to follow the voice. And that shiny little object, or fluffy object, I guess. So we learn our body scheme. We learn our reflex maturity, how to coordinate our reflexes, our ability to screen input. What's poopy, like Professor Yari Harari has said, and then what's real. Postural adjustment. Then we move, and if I've been sitting like this at the screen, which happens, then I just like, oh, I got to change that. Ocular motor control. So we organize how this works so we can look off there and there's a sound coming from that direction. I'm going to move my eyes and eventually I'm going to turn my head. But if the muscles aren't working or your neck is messed up because you've had 5 car accidents, turning your head can be a challenge. Eye-hand coordination, teachers know this. If a kid is 5 years old and they can't use a pair of scissors, they got to start working on the things down here. And it all goes up to the sanayog, the cognition or the true self. Boy, we covered that really fast, didn't we? Let's go back to Ardem. Oh, poor Ardem. I'm sorry. He's a fantastic individual. And it's necessary for us to kind of dive into it. So I decided to, nope, that's not sharing. I decided that it was easier to show you the different screens that I've taken. And that way I won't be clicking on his play button. And so, okay, again, we're talking about olfaction, taste, vision, hearing, touch, and pain, coupling together in order to help us with our perception of reality. And he says at 3 minutes, 22 seconds, he is here to tell you that your sense of touch is amazing. Human tactile discrimination extends into the nanoscale. And boy, do we have nanoscale. Neil Thies and his group in July 2025 had the paper published about the reticular network in the pancreas and the interstitial fluid filled spaces. Brilliant. So it's extending into the nanoscale, and these nanoscales are all helping us to understand information. Well, input information so that then the body can process it and understand it. And we're digressing, but Alex Chesler wrote a paper there it is He wrote a paper with Marcin Secot, and Marcin is working with the NIH, but also Linköping University. And you can tell this is not a last name that comes from Sweden. It's not a Johansson or a Pete Pettersson. So Marcin is an immigrant here learning about touch, and I think he's in Linköping. Yeah. They say piezo ion channels are trimers, sorry, are trimers. So tri. And they are shaped like a propeller with 3 blades organized around a central core. And I think that's important because when we go back to Ardem and he discusses with us about piezos, gotta put you on hold. Can't find it. It's just one of those things. I have a lot of screens up and you don't know how many, but I have to find the right thing. But Ardem says this extends into the nanoscale so that we can detect we can sense indentations 500 times thinner than the human hair beyond. Yeah, we rely on this affective, it's spelled wrong, affective, AFF, affective neurons. That's what Håkan Olsson and Francis McClone were talking about, and that's slow touch. And keep in mind, Alex Chesler, we just saw with that hair pull, that lasso receptor that's lassoed around the hair follicle, that doesn't respond to slow touch. It responds to something heavy. So afferent is more of it's the emotion side of things, I think. Me. Whereas our affective is more of the behavioral side of things. So the information going up affects our emotions. The information coming down is more of our behavior. That's the way I've put it together. So this touch is allowing us to know where our fingers and arms are into space, but it's helping us complete important, critical, complex tasks, such as walking or playing a musical instrument. So he does show
walking.
Yeah, he does show us how kittens love to snuggle together, and that is their way of receiving information. This is their own little hug, and this discriminative touch we have on our fingertips is so critical to how we learn to play musical instruments. And because we do it again and again and we train, we can do it without thought. And eventually you can have 2 masters rifting together, and they create something extremely complex and beautiful. So how
side of touch is, of course.
So I have to share the next video screen because I don't want to have too much of the audio come on because it will copyright. So let's see. Yeah. So at 4 minutes and 53 seconds, he's Dr. Pataputian, Professor Pataputian says, our environment is essential for sensation of our environment is essential for survival, period. Chronic pain, when the noxious stimulus is no longer there, but pain persists, remains a major unmet medical need. And that's why Lorimer Moseley and the whole group in Australia that's talking about pain and reprocessing, reprogramming the body to understand something is critical to learn how to deal with pain. But here is a man saying, we have another back door. we have a way of opening up these piezo channels in a way, touch perception. And he goes on and discusses the neurons responsible for detecting mechanosensory stimuli relevant for touch, proprioception, and pain. Mainly, this resides in the dorsal root ganglia, or DRGs, a group of sensory neurons present just dorsal to the spinal cord. They gather information from the periphery and send the information to the spinal cord and then to the brain. So that brings us to somebody that we all know, Paul Hodges. Where's Paul Hodges from? Australia. Yeah. Super. Yeah, we're getting there. We're so getting there. So Paul Hodges on day 2, along with Frank Willard, an osteopath, did a panel discussion, and the morning speaker at the 5th International Research Congress in Berlin was chatting. And so this is a perfect example that we have information here that comes in, and muscles pick up that sensory information and send it to the DRGs, reflex spinal networks, so those sensory neurons, and then it goes up to all this processing. Man, that's a very complex. You can't see that. The motor system involves an enormous amount of your nervous system. All nervous system regions are involved in generating, planning, performing movement, sensing the position of the body and how to move in its environment. This is proprioception. This is a sense. We need it to get up to here. Unless you're Ian Waterman and you relearn. Muscle is really important. Muscle is the way that your nervous system interacts with the environment. You interact by motor commands, causing muscle contraction, leading to movement. If the muscle is unhealthy, it will compromise all the ways your nervous system has the capacity to interact with the environment. And Ian Waterman had to figure out, he had all the downstream information for motor coordination. What he didn't have was this upstream sensory information throughout his body to tell him how a cup felt, to tell him that his fingers were feeling change in his pocket. He had to hear the change in his pocket. And he did it. At 19 years old, he lost the sense of touch. He retaught himself, but it was a painful process. It was a long process. And Mandy Harvey, same thing. So Mandy Harvey, as we saw on the Golden Buzzer, America's Got Talent, Mandy says to Howie, one of the judges, she says, yes, I lost my hearing when I was 18 years old. So at 4 years old, she starts to learn how to play musical instruments, how to sense cadence and vibration and tone and everything. So she has all these bucket brigades working its way up. And as we know from Donald Hebb, neurons that fire together wire together. And Donald Hebb, fantastic researcher and scientist about behavior, Mandy had to relearn how to feel the vibration in the floor, just like elephants do in their seismic communication, because she takes her shoes off and she feels the vibration in the ground in order to get the right tune, the right pitch. And if you have a chance to go on and listen to Mandy Harvey, fantastic. Hey, Siri, play Mandy Harvey, Try. Here's Try by Mandy Harvey. God is great, Lord, that but I know one day I'll get and don't take my place. Go around. Hey Siri, turn off. I mean, that's gorgeous, isn't it? That's gorgeous. It's amazing. So many branches, so many linear thoughts. Let's come back to the task at hand, Ardem. Those piezo's, piezo 1 and its sister pressure receptor, ion channel piezo 2. So important for how we sense the world around us. And it's happening at the nanoscale because, as you saw from Alex Chesler's paper, those trimers, it's like a fan blade. Think of a fan blade, and those blades are all sitting together in threes, and then they pull apart and they open, and all these ions flow in, millions of ions in split seconds to create an electrical change, voltage. And then that voltage then gives us chemical responses. And as Ardem brilliantly says it, it translates it into information the body can understand. And this all happens without our conscious thought. And this all happens because we are given this incredible natural body that already gets it. It's nature, it adapts all on its own. But we can help it, and we can help people in pain, and we can help emotional dysregulation because this is because touch is the foundation. And what we remember from Francis McClone and his back in the sack baby mattress for neonates, it's changing their whole physiology when they get that slow touch system, because that slow touch system goes up into the brain and it's processed in the same part as vision. How incredible. We wouldn't know this without microneurography. We wouldn't know this without Oke Valbo working his whole life trying to figure this out. And thank you, Håkan. Maybe you'll get this video letter. And thank you, Laura Case. Maybe you'll get this video letter. But without those researchers, without those scientists, we wouldn't be here today talking about how touch is so important for our emotional regulation. And we all know from COVID and when we are locked away, except Sweden, we did things differently. And we got a lot of flack for it. But there was so much high anxiety. And that high anxiety it was paramount throughout the whole world because everything changed and it was hard to grasp. And so that emotional dysregulation, it was just this vibration throughout. Now, a virus caused COVID, and we know how to protect ourselves from viruses now. But the thing is, some people hang on to viruses longer. And Ian Crozier is one of those individuals. Now, Ian Crozier was a doctor that went over to, I think, Sierra Leone in Africa to help fight the Ebola virus. So we're digressing a lot from Ardem, but this is important. So Ian, let's see. Ian Crozier, after nearly claiming his life, Ebola lurked in the doctor's eye, Emory Eye Center, and then you have the New York Times article by Denise Grady. So that was May 7th, 2015. So Dr. Ian Crozier, 44 years old, ruthfully calls himself a poster child for post-Ebola syndrome, long COVID. Besides the eye trouble, he had debilitating joint and muscle pain, deep fatigue, and hearing loss. So Ian Crozier went as a doctor. He had two blue eyes. after he was told he was cured of the disease that he had, Ebola, his left eye turned green. And so there's all sorts of information here, but both these doctors volunteered in Sierra Leone. And the main problem is that there are lots of body and joint pains, chronic headaches, and women who stop having their menstrual periods that have had Ebola. There's quite a bit of vision problems as well. He's met one former patient who does appear to be deaf. The hearing loss could result from the brain inflammation or very low blood pressure for an extended period, both caused by Ebola. Now, the nice thing is, Ian Crozier got better, but he was told with a blood test that he had no longer had the Ebola virus in his body. But the problem is, 2 months later, he was back in the hospital because his eyesight and intense pain and blood pressure were, and the soaring pressure in his left eye was, he couldn't handle it. The pain was so bad. There is something wrong. And the results inside of Dr. Crozier's eyes was teeming with Ebola. And the doctors were amazed. They had not considered the possibility that the virus that was in his blood and in his body had actually invaded his eye. And months passed. And finally, they solved the problem. And he had uveitis, and Dr. Crozier's eyes were examined on March 27th at the Emory Center, and eye troubles were common among Ebola survivors. And what we need to know and understand is that Ebola, a virus, just like COVID, a virus, we can test for it in the blood, not there. But what we know from the eye is a mini interstitial fluid filled space, the interstitium. Did not know I was going here today, but here we go. I spent a lot of time on this the other day, so I will not bore you with all the details, but in my sense of reading Neil and others, see lots of people, Rebecca Wells as well, the evidence of interstitial continuity within and beyond the human pancreas that has everything to do with the perineurium, has everything to do with cardiovascular health, is these IS. It's the interstitial fluid-filled spaces. And fluid-filled spaces, IS, are small, but they're numerous, just like the piezo's 1 and 2. They are so super tiny, but they're vast. And when they couple and they synchronize and thousands of them open up in every second, we get voltage changes. We get chemical responses. We get information that goes all the way up the bucket brigade to be processed. And so when I say that this great information from Neil and his team, that the interstitial spaces contain hyaluronin and collagens, and most appear to contain elastin, proteoglycans, and a variety of glycosamines. So hyaluronin is highly hygroscopic, which I did not know that the first time I read it. Hygroscopic is adhesive and movable at the same time. I kind of think of it as cornstarch in water. So if you use a slow pressure, you sink into the cornstarch. If you use a hard pressure, then you hit really fast. And to me, that is the definition and the visual of hygroscopic. So all the interstitial fluid-filled spaces are continuous within organs, whether it be the skin, colon, liver, and between adjacent tissues, and underlying subcutaneous fat, as well as the colon and subadjacent mesentery. And here's the thing, this is all governed by receiving information from the DRGs that richly, robustly innervate fat. That's the next thing we'll get to. But first, we have to continue on this particular line of thought. And the space, I should actually say this was July 4th, yesterday, when I put the finishing touches on it. So vision is to the eye, so critical, so important. 126 million photo transducers, whether that be cone or rods, and Ardem Pataputian's Nobel Prize lecture, you're going to find out the whole history of Nobel Prize winners that have understood the sense of vision, have understood that rhodopsin in the sense of vision is also important for the sense of smell, another Nobel Prize winning. And this is all compact in Ardem's Nobel Prize lecture. So the eyeball, and as an example of and similar to the description in the July 25 paper, evidence of interstitial continuity within and beyond the human pancreas. So the eyeball is a perfect micro example because as interstitial fluid filled spaces are defined, they can measure up to 100 micrograms. They contain glycosamine glycans. They contain hyaluronic acid. So what do we have in these small chambers and clear fluids of the eye? It gives us a sense of vision. It's the structure and fluids that play a key role in our sense of vision. And parts of the eye have to work together. And the funny thing is that I chuckle about is because Steve said that in the Leap about Mars, he was even surprised they even got to Florida. I mean, let alone sending rovers to Mars, that they just actually, between failure and failure and failure, that they actually were allowed to go into Florida and launch these. But because a team of scientists and engineers worked together as a unit and they coupled all of their knowledge, they were able to do it. And here's the thing. This fluid in the eye carries oxygen and nutrients. The fluid plays a part in the eye's immune defenses. And that's why Ian Crozier also had a problem with the Ebola virus, because the fluid is in the chamber spaces are full of hyaluronin. And hyaluronin, just like the fluid filled space here, is innate. What do I mean by that? We're born with it. It is something the immune system does not recognize as being a problem most often, unless you're immunocompromised. But these fiber networks are a large scale interstitial, if there's fiber networks. So this eye has collagen in it. It has a fibrous network, but it also has this fluid filled space with glycosamine glycan and hyaluronic acid, hyaluronin. So the right amount of pressure has to be in the aqueous humor, which is a fluid, in the anterior chamber, in order for us not to have high pressure or low pressure glaucoma. And the pressure gives shape to the eye. So we have the virtuous, which is glassy, chamber, which is a space, in the behind space cavity. And this is one of two spaces. And this space is filled with a gel, and it's between the lens and the retina. Very important. And the posterior chamber is an important structure to help produce and circulate aqueous humor, which I'm not sure if it was aqueous or if it's, yeah, sorry. This should be vitreous humor. That's the posterior cavity. Now we have an anterior cavity. It's called something. But when the anterior cavity is a fluid-filled space inside the eye between the iris and the corona, the innermost surface, the endothelium can be irritated. And we have three major types of pathologies. Hyphema, don't know anything about that. Anterior uveitis, that was Ian Crozier, and his eye color changed because the Ebola virus was living there, and it was really painful. And it also can cause glaucoma. And glaucoma is the blockage of trabecular meshwork. And this prevents the normal outflow of aqueous humor. So therefore, too much pressure, too much turgid pressure. And I use turgid pressure because plants have a turgid pressure system. And that moves their xylem from the roots to shoots, their water and minerals, and their phylum from the shoots, from the leaves to the whole. And it's food made by their photosynthesis. Their leaves photosynthesize all this carbohydrate, and it makes their own food, and it goes throughout the whole plant. So nature has found a way. I go through so much here. You can absolutely just dive right in. But the vitreous humor between the lens and the retina, yes, lens and the retina, makes up 4/5 of the volume of the eyeball. And 4/5 is 80%. 80% of the volume is this fluid filled pressure, fluid like near the center and gel like near the edges. It contains hyaluronan, small amounts of sulfated glycosamine glycans, CS, I don't know, couldn't find it, and heparan sulfate. So where did I get all this information? There's my sources. Wikipedia, Cleveland Clinic, a Nature article. So we have to have regular checkups for our sense of vision. And we check the aqueous fluid. And why do we not have regular checkups for our interstitial fluid filled spaces? Well, we don't recognize them as important until Neil Thies came along and worked with his team, Rebecca Wells and more. And it was Neil's curiosity of having an endoscope, because he's a liver pathologist and looks at dry dissected cancer specimens, specimens underneath a microscope. They were all collapsed structures. And what he found out is that collapse is, he was told in his training that that's an artifact of the dissection process of being dry. But what he found out when he put the endoscope on his skin, in the living body, they're not dry. They're filled, those chambers and those spaces and those cavities are filled with fluid. And fluid, just like a plant, has to flow. And if the pressure's too low, we have a wilted plant. If the pressure's too high, we have a drowned plant. So what happens to a wilted plant? We give a little bit more water and it perks right up. So if we give a little bit more water and we help the fluid flow and we change the pressure inside, not only does our eyesight get better, my shape of my eyeball probably hasn't changed, but I use a tool to help my sense of vision. And I think touch can be the tool to help the body learn a new processing or help these receptors keep being stimulated. I've gone a long ways. We've gone a long ways. We're going to come back. We're going to go back to the original task, the whole reason I started this. The last video we do, video letter of chronic pain and neurodegeneration, is the last one. I could go in so many different directions, but it's important that that will be the last one. This one is ending. Just to let you know, we're ending very soon. So Ardem talks at the Chinese Brain Conference in 2022, Beijing Brain Conference. And he says PZO1, what they have found in his lab is mechanosensitive. It's also in blood and lymphatic vessels. It affects bone. It affects the health of skin. It affects red and white blood cells. And PZO1 is expressed in bone. It helps bone form and regulate bone density. Could osteoporosis, osteopenia be actually an input penia? Sarcopenia, muscles. Could that actually be an input penia? Maybe these PZOs are turned off. Maybe they just need a refresher or to be trained. I don't know. It's all a question. That's why I'm writing this letter, Robert and Andreas. Oh, that's why I'm videoing this letter. Astronauts in microgravity without gravitational pressure on the bones, they lose bone density. PZO1 may be involved in the process. So we have more. Oh, do we love Ardem? We do. We do. We do. We do. So he also discusses at 16 minutes and 56 seconds Alec Chesler and Carsten Bonemann's work at the NIH when they identified people that were lacking, brist is Swedish, lacking PZO2. They had to do gene sequencing. Many times, as Dr. Ardem Pataputian says in one of his Scripps lecture from 2018, that many of these individuals were said to have movement disorders, when in reality, they have a sensor processing disorder. Their genes were sequenced to find out they did not have a PZO2. So PZO2 deficient humans lack touch, sensitivity, and proprioception. So this woman, Chesler, in 2016, I think, Chesler and Bonemann, they did this at the NIH labs. This is a female college student, and I think she's an engineering major. And she did not know that she was lacking any genes because somehow along the way, her body figured it out all on its own. It learned with the lack of, at a very early age, how to navigate in the world. But like Ian, if she doesn't have her sense of vision, she's lost. And she can't even find her nose with her finger. But when she uses her vision, she can do this. When she uses her vision, she can walk. Here, she doesn't have the sense of vision, and that's why they have her in the harness. So they play this video and she tries to walk and it looks like she's drunk. It's not a movement disorder. It's a sensor processing disorder because of input penia. You can tell I get a little emotional when I do this because I think this is all coming together. I get excited. Where are we going now? Oh, we still have more to talk about. Dr. Ardem Pataputian was on the TV show Vetenskapsvarld, which is Science World in Sweden, and they talked about the climate crisis. They talked about the Schensel feeling, which is touch, and the chemosensation. And we talk about this in the functional atlas. Well, Carla talks about it. And there's all sorts of fascia books. This doesn't talk about fascia, but it talks about the bone and the ligaments and the skin, all PZO1 innervated. So Ardem is asked at the Beijing conference. I'm trying to find the window on the screen. So the Chinese, I just lost my word, but the person organizing the conference. And what inspired you to explore touch? And in the developmental biology of DRG neurons, dorsal root ganglion neurons, I was fascinated how some dorsal root ganglion neurons become different. Some neurons project to muscle and become proprioceptors. Others become free nerve endings, but some sense temperature, others sense, in some sense, other. The vast heterogeneity, what these neurons do is extremely unique and that other neurons do not do. Sense physical stimuli such as cold, heat, and mechanical stimulation. And if we don't have these, and if we lose them, if we don't keep training them, if we have them, or we don't learn a new, different way, we have a problem with longevity and health and how we feel. You guys can see it, but I have to get all these windows that I have up down there. Yeah. Ardem asks at the Beijing Brain Conference at 36 minutes and 18 seconds. They're still doing research in his lab. Now, given what's happening in the United States at this time, his lab may not have funding. So you being in Germany and hopefully Håkan being in Sweden will help progress this research on PZO1, PZO2, emotional regulation, and how important it is for pain, because it is a major unmet medical need. So it's unpublished data that he shares. Sorry. The agonists are unknown, but we know that body work stimulates the parasympathetic nervous system. And maybe what the body work is doing is helping that sympathetic nervous system, that fight or flight nervous system that really gets excited in times of stress. Maybe what we can do is to bring them into allostasis. And we have this load here for sympathetic that highly innervates sympathetic, the superficial fascia. And we can bring them into allostasis. So the load is not so great on one side. We talk about balance, but I don't think things balance ever in the world. There's never really a time of peace. So Ardem asks, why is there a mechanosensor sensing fat tissue? Well, many sensory neurons express pressure-sensitive PZO2. The dorsal root ganglion neurons robustly innervate fat. What is superficial fascia? It's that subcutaneous fat. We need it for all of this to work. Surprisingly, the dorsal root ganglion has the brake on the sympathetic nervous system, and it regulates adipose function. Similar to blood vessels of baroreceptors on the vagal system. Stephen Porges can talk about this at length, obviously, in August in New Orleans. And so the unknowns are the agonists. So sympathetic tone may change, but how do we do that? I guess that's a question du jour. It is. I have something really cool to show you. I'm excited because, oh my goodness, we're wrapping up. We're wrapping up. We are wrapping up. We're coming back. We're coming back to this task that's been sitting right here. Yes. So now we're going back to Ardem's lecture at the Nobel Prize Award. And he says here the skin is responsible for the sense of touch, and he is here to simplify sensory receptors responsible for touch. And there are different fiber types, such as Merkel cells and Meissner corpuscles, and they play unique roles for our sense of touch. And if you remember, it was here in this book that we discussed in one of the video lectures, how the Meissner corpuscles reduce in density and quantity as people get older. And the older age, the 70 to the 80-year-olds had 75% less Meissner corpuscles. And that is a hard thing because just like the lady with the match, it took her 20 seconds extra just to light a match when she couldn't feel those three fingers. And that's research from Roland Jansson's lab up in Umeå. Yeah, we have lots of cool things that happen, but we have to understand it. So Ellen Lumpkin and David Gitney help an awful lot to the understanding this process, but also in addition, many DRG, dorsal root ganglion neurons, project to internal organs such as the bladder, and they're involved in a process called interoception. And yes, Ardem talks about that later. Woo! But at 10 minutes,
family of GPC.
yep. So there he's talking about vitamin A and olfaction, and that's the history of Nobel Prizes that were awarded. And you know, any time of this research, this is 10 to 20 years. So PZO1 and PZO2 was started in 2010. What? No, wait, sorry. They found them in 2010. And now we're in 2025, 15 years later, and we're still not talking about it in the doctor's offices. And so something has to change. And Brian Pulling, who went to Lorimer Mosley's pain schools and Natasha Stanton's pain group, hadn't heard about fascia. And for me, somebody that's a pain researcher like Brian Pulling and helping people, it's not only about muscle. It's not only about doing silly exercises on a piece of paper. It's about touching that person and making sure that we're training these mechanoreceptors, which are they're sonic pressure sensors. Sonic, sound wave. Pressure wave. Okay. PZO1. Yep. So I think it's really important to find out. There's a young Ardem Pataputian, but 2010, the faculty member Marcel France Beltron, he found a way to assess mechanically activated channels so that he could record them from a cell. So a long process, crazy, a reductionist approach, found out many different things, and it's playing a critical role. And so he tried different cell lines, 300 different candidates. But where am I going with this? 1425. Oh, yeah, it's the picture of these PZO1s. Holy crap. So they have a sister. And look at all these little ribbons. I mean, this is nanoscale. Nano. So he talks about the process of how they are talking to the plasma membrane of the cell, and that tiny tension and stretch of the membrane will cause a flattening of these structures and hence activation. It's critical that we have touch. And what I can say is if you have ever stubbed your toe or you have a child that comes to you that has fallen down, the instinct, reflex, whatever you want to call it, is we touch it. So soft, slow touch eases our emotion. There's a reason we touch. There's a reason we hug. And if a child comes to us and has a skin their knee, we care for them. And we have to, I hate the word should, have to, yeah, because life is crazy and it has a lot of things going on. But if we can teach people how to take care of their own bodies, how their bodies can adapt all on their own. This is critical for well-being, emotional regulation, and the sense of pain. So calm down those nociceptors. Give them their multi-personality. So give them back their proprioception. Give them back their sense of touch, not pain. PZO2 is robustly expressed in the sensory neurons of the dorsal root ganglion. And these are the neurons he told us about that were required for touch and pain. And this is this, don't know. So they made transgenic mice, which means they created a mouse that did not have PZOs. And they can do that with mice because that's what scientists do. So the sense of where your body is in space is proprioception, and it's based on mechanosensory neurons that innervate muscle and tendons. Thanks to the sense, they're all coordinated, they couple, and you can close your eyes and touch your nose, unless you don't have PZO2. So PZO2 is required for proprioception. So in collaboration with Wilkinson and Jessa, they made this transgenic mouse that does not have PZO2. And here you see the mid-thoracic area of the mouse. And this mouse lacks coordination in its hind legs, a sign of lacking or being brisked or deficient in PZO2. Therefore, if you're deficient in PZO2, you're deficient in proprioception. And just like the college engineering student, she did not know, because at an early age, she was born without it, right? She figured her way out. But my mom, being in her 70s and 60s, 70s, and 80s, did not know that these were diminishing. And I tried to get gene sequencing. And unfortunately, for the average person, there is no gene sequencing for these TRP receptors in PZO. There's just not. And that's a shame, because therefore she wasn't treated the correct way. She was told that she had a movement disorder. In a sense, she had a sensory processing deficit. So, yeah. Oh, I know. It's hard to see. It's hard to see. It's really hard to see. Yeah. And then 1720 to 1716, we did that. 1722. 1722. Yeah. So you can see that this mouse lacks coordination. Interesting. They have the same deficits in touch and proprioception. Oh, yeah, here's the engineering student. Da, da, da. I knew it was somewhere around there. Yeah. Oh, let's wrap up. Where did that screen go? There it is. Patricia Houston? Houston. Hudson. Houston. Can't remember. Patricia, I'm sorry. You were on one of the biotensegrity calls, and you were kind enough to come in and chat with us. It's when Mariana Barato was talking about her cerebral palsy children that she works with sorry, a manual therapy called ABR, and Leonid. I'm sorry, I forgot your last name. He's from Poland. And no. I'm sorry. I think you're Ukrainian or Russian. I apologize. But he's got a very loud voice. And he's been to all the fascial conferences. But he has ABR in order to help these cerebral palsy children that are born most likely, in my estimation, with a low amount of adipose tissue. So therefore, their sensory neurons what are they going to innervate because there's not enough adipose tissue? And it seems that from the videos that Mariana showed, when these children were worked with at a young age, their body morphology changed. It became more voluminous. You can even see the before and after pictures after work with ABR. These children were learning to thrive, not because their body couldn't before, it's just they didn't have the right stimulation. They had an input penia. For some reason, their body hadn't gotten enough adipose tissue. And so with the work with ABR, Mariana and Leonoid are able to help these children. It's brilliant. It's another intervention. Before that, it was only surgical interventions. Anyway, Patricia was there and she came up with this paper written in March of 2022. It's a great paper. Chronic disease progression, changing interstitial cell behavior. And if we can change interstitial cell behavior, it's so much easier for the body to make the decision on its own. And when we have the body make a decision on its own, like Professor Yuval Harari says, we can help the emotional regulation, and we need to teach our children how to emotionally regulate at a young age so that they can form better reactions. And the emotion is what separates us from artificial intelligence, which is a non-organic agent that can learn crazy amounts of things that AI can actually not only learn, but make up new stuff because of all this collective information that they've gathered from various different disparate sources, just like this one. Their sensor fusion sensor fusion and AI is crazy involved, but this is too. And our whole body is a sensor fusion, and we can help children and we can help people be better. Like Patricia says, we can, what does Patricia say? Well, you saw the picture. My brain is frying a little bit. I'm sorry. What did I just I can tell that I need some food because I'm going a little bit on yeah, uh it's almost 11 o'clock. This is way too long of a letter. I'm so sorry. But what I was saying is Patricia's information is changing interstitial cell behavior. And how do we do that? It's the same thing that I just talked about with Mariana. You know, Mariana Barreto, her cerebral palsy children. And I wrote her and, sorry, it's fast. I wrote her and Leonoid Bloom, Advanced Biomechanical Rehabilitation. Yes. So I wrote them after they were, oh, sorry, that's too fast. They were on Big for physios, Saturday, March 23rd, 2024. So Mariana did a presentation and she showed visuals of cerebral palsy children. And is it a physical or a sensory issue? And the reason that I think it's a sensory issue is because their advanced biomechanical rehabilitation is offering mechanotransduction. It's exactly what Tricia just said. Cell behavior is changing because a stimulus is offered to open ion channels, which change their signaling capabilities, and their behavior changes. Therefore, the behavior in the bucket group grade all the way up changes. But dorsal root ganglion neurons of the peripheral system are stimulated, and they are the ones that are innervating adipose tissue. And that's why her children are having better fat distribution and density. And that's why they have enhanced hormone secretion. And that's why they have self-regulated phosphorylation. And that's why their cells can form and they have shape change, and therefore ligands can bind to it. And if they activate or inactivate the transmembrane receptor of with new ligands, it's better for their whole physiology. Dorsal root ganglion neurons are found in the peripheral nervous system. They innervate skin, muscle, and internal organs on the macro scale, and they gather information or sense temperature and force of these structures in the periphery. And that came from Ardem Pataputian, The Forces with You, December 2018. Yeah, my husband's calling. I've talked about this book by Dr. Joan Vernikos. And again, you'd need to know the backstory, but she grew up in Egypt and her father was a doctor. And at the time, the way that they found out somebody was diabetic is they put their urine on a plate and they set it out. And if flies went to the urine because it was full of sugar, then they knew the person was diabetic. There were ways that we did things that we have totally forgotten. But she grew up being curious, and she went to NASA and she saw a need and she became their doctor for stress handling, because astronauts are trained. But you know what? Being an astronaut is very hard on the body because every system in the body changes being an astronaut. So we are going to go to some of her information, because she says in her book on page 48 is that minimizing bone loss and protecting the joints from wear and tear are essential goals for anybody concerned about health and active aging. But there's no magic bullet, no single treatment. It is an absolute answer. But she says preserving collagen is critical. Robert, I know you're going to like that. Preserving collagen is critical. And then she talks about loading omission. Now, loading omission in gravity, you can, as an astronaut, you can work out every single day, but there's still changes to the heart. There's still changes to the cardiovascular system. And without the working out, the changes would be worse. But we're going to go to one of her talks now and talk about it. But this loading omission, if we don't mechanically load the body, just like Carla Esteco talked about in her book, the deep retinaculum is not in fetuses. It develops as we mechanically and load the body. So let's just dive in, because Dr. Vernikos is fantastic when she talks about this. So I have a few things. This is her. This is the transcript. This is how we're going to do it, because I don't want to play this and then have it copy struck for copyright. We're going to go to 2231. She tells everything here. 2231, 2231, 2231. Okay. So,
you're not lazy.
This is what I didn't want. Hang on. We'll get there. Balance and coordination. We totally forget that we were this age and we learned to walk all on our own. But the feet stand wide apart. And what she says is astronauts that come back from space also take this same stance, so they have a wider base and they can stabilize themselves. And the next thing she talks about is Rick Seaforce.
So Rick
yeah. So the transcript says Rick CFO, Seaforce, SEARFOSS, I believe is how it's spelled. But Rick was on many shuttle missions and space station missions, and he had just returned from a 9-day shuttle flight And this was in 1998 in May. So he was being tested on NASA's posturography testing, which we don't have. My mom never had a posturography test, and older people don't have posturography tests as a standard test. We have vision tests, we have all sorts of other tests, but we don't have a posturography test that would tell us something. And here's the thing, Rick was on this balance board and something had happened, and she's standing beside him and everyone's chit-chatting, and then all of a sudden she sees Rick sway on this balance board, and he can't keep his balance, and he starts leaning and leaning. Now he's a big guy, and there's a lot of people around that rescued him because he did not realize he was falling forward. He couldn't even stop himself. And that's where my mom was in her movement disorder. But as we see with Rick and other astronauts, they come back from having this loading omission, being in microgravity, without the ability for their body to sense gravity. And there's all sorts of things that happen. So let's go to astronauts lose bone in space. 14. So we're going backwards. This is part of my thinking process. Okay, so there was some polio studies by this man, a friend of hers named Don, and they used these polio studies to find out on people with bed rest and polio that they also discovered that astronauts lose bone in space. So using one discovery about polio, they applied it to the astronauts and they said, yes, they're losing bone. And it's inactivity is one thing, but it turned out that the same thing about lying in bed that makes you sick in healthy people is also the bed rest, and lying in bed is the same thing as loading omission that happens in then astronauts. And without that gravity and that force, the pressure of gravity on the body starts breaking down. And so, yes, we have gravity here on Earth, but if you're in a sedentary position, if you're in bed rest, you have a loading omission. Inputpenia is, yeah, like osteopenia, sarcopenia. And it leads to dysregulation. So let's go to the Russians. And at one time, scientists learned from each other. And the Russian cosmonauts were spending a lot of time in space, and they were going longer and longer. In the 1980s, they were spending up to 6 months away from Earth. And it was hard for them to get a good night's sleep because they were complaining as if when they were lying in the bed on Earth, that they were sliding off. Their proprioception, their vestibular system, everything was screwed up. They're lying still, but they felt the bed or their body was moving off of it. That's weird. Of course you can't sleep. So when they talked about this, the Russians and I think the Americans, they figured some things out, and they did it together. So they found out if the bed was horizontal, it was harder for them to get a sense of position. So if their feet was on the ground, it was better, and they wouldn't feel like they were slipping off. But then they found out they found that lying head down in 6-degree head down position was just enough in this bed rest model that astronauts returning from space and long space could actually get a good night's sleep. I don't know how this changes the body, but we talked about pressure systems, and we talked about interocular pressure causing glaucoma and blindness eventually because of the pressure is just not right. But maybe somehow this changes our pressure system because all these mechanoreceptors, these sonic sensory pressure sensors, maybe that changes it just enough. So that was good. That was very good, but it's thinking outside the box. It wasn't just inactivity, it was effective reduced gravity, changing these receptors because the body adapts all the time. So then we'll go over to the 9-day trip for Rick in space. Rick Seifers changed his body enough that he could not propriocept and be on a balance ball. But as we see in 2025, '24, 2402.
Meet sauna.
Okay, it won't do this. Every system in the body is affected, and astronauts have insulin resistance after being in microgravity, muscle atrophy, bone loss. The heart is smaller, and that means it pumps less and it's less effective in its reduced heart rate. The stroke volume and the transit time in the gut is also slower in astronauts, and everything, absolutely everything, is changed. The muscle atrophy is followed by fat moving into the muscle, into the atrophied muscle. Okay. We talk about fat moving into muscle, but the quickie, the takeaway here is the DRG neurons robustly innervate fat. So if the body can't sense, or if the DRG neurons are getting input penia, maybe, perhaps maybe, the body's defense system is to lay down more fat. That would be a really cool way to adapt, wouldn't it? So just putting it out there. Well, how is it that these astronauts get insulin resistance with being in microgravity? That has nothing to do with their diet. So we're going to go back to Joan because I got rid of that silly advertisement. This image is taken, and again, in her book, she talks about the research that goes into this, but this image is taken because animals that went up to a 9-day mission or a 10-day mission up in space, they were sacrificed, and so we can see what's going on in their endothelial lining. And the image is the change in space that happens. The lining of the blood vessels, the normal ones are on the right. The middle and the right are different subsections of the blood vessel. So these two are the same, are in healthy tissues, healthy blood vessels. So the lining around the endothelium is stimulated by shear force. So we have proper blood flow through here. But if your heart is smaller and the stroke volume is smaller, you don't have the same amount of blood flowing through the blood vessels. Now, shear force of blood moving through this vessel stimulates the endothelium to make a substance called nitric oxide. And it happens to be something very important because it helps make blood vessels constrict easier and expand easier, and constrict and expand. Therefore, we have a better sense of, well, our heart works better. So animals that flew in space for 10 days is here. And you can see the normal blood vessels, what they should be. And eventually the lining, after 10 days, the lining in this animal of the endothelium is changing. And if the animals have been in space long enough, it disappears completely. And it's not just in one area, it's throughout the whole body. So serious changes happen in space. Now, astronauts recover, and that is a great thing. But if we have multiple layers and multiple receptors that have input penia, none of these little critters in the ecosystem work well or anything. And eventually, something like this with the Danish handball player, her pressure of her body absorbs the hit. And eventually, she won't be able to do this in the older ages because the hyaluronan and that absorption system, that shock absorption system, is no longer working. She's young. She has a lot of hyaluronan. She's robust. She's fit. But like the astronauts, we can work out for hours on end. But if we don't change this system, and this is for our friend J.C. Gumbelto, if we don't keep this wet and hydrated and these interstitial fluid-filled spaces filled and our pressure just right, we're no longer dewy, we're dry. Not a good situation, is it? No, no, no. So an elephant, we know from Caitlin O'Connell's team, for after 20 years of research, they have seismic communication. All these vibrations in the dense ground substance, these sonic pressure waves, these vibrations send a signal from their fat pads in their feet to their brain so they can decide something, so they can react, so they can make a decision, so they can survive and they can thrive. And we know from Danielle, who is now named Danny, because she was adopted by a wonderful man in life who had a lot of challenges with this situation. And eventually they divorced, but the father, he still visits Danny, and in the home that she's in, and she has this pharmaceutical cocktail that keeps her basically upright. But because of her early life experiences and her her sensory nervous system having to find a way to survive and thrive, it did it. It adapted, but at the cost of not knowing how to graft, at the cost of not knowing how to brush her hair, at the cost of not knowing that a dirty diaper is not a good thing, and at the cost of never being able to hold a fork at a young age. So eating is a little bit of a challenge, and she doesn't eat like you and I. And she has learned, but it's taken a long time. So it's not all about pharmaceutical interventions. It's not only about surgical interventions. It's about making sure this system of acoustic baffles that's throughout our body is either absorbing signals so they're not so noisy, or it's making sure that the bucket brigade is sending up the right signals. Yeah. So Joan Vercanos just shared with us about nitric oxide, and in this particular collection of papers, we go to Denise Popping and Ingrid Sarlis, and it's about extracellular, I gotta read it, extracellular matrix fibronectin mechanically couples. There's the word, couples. Skeletal muscle contraction with local vasodilation. I'm reading it backwards and upside down, but I can do it. And this is a great paper, because nitric oxide is produced in our nose, our turbines, and they spin just like that piezo. They spin and they heat up. They spin up. Their speed changes the air so it warms, because the speed of sound, it changes in higher temperatures. And you want to know something really cool, Robert? This is the cool thing. Nitric oxide discussion at the British Fascial Symposium in 2024 in the panel discussion. Leon Chatow is there, Sari Davis, who was facilitated by Julian Baker. Thank you, Julian, for all your stuff that you do. Leon Chatow says, nitric oxide is also stimulated by the vibration caused by humming. And Robert, you're sitting right beside him, and you just learned something. And I have this face all the time. And if we dare and we relearn something that we learned previously, but we have to relearn it,
or we learn something new, this is that light bulb moment, isn't it? Yeah. I just thought it was fun. I just had to share. Yeah. But as we have discussed, the somatosensory system, even skin receptors, if they're changing, there's a decline. If there's a decline in tactile stimulation, it's all about that microenvironment, that environment, just like the bugs on the hands. Inputpenia and Mark's paper. Mark's paper, other papers that he has, pretty intense about the tribosystems of the body, but all of the sliding and gliding that happens inside the fascia, it has to be just right. It changes pressure, it changes fluid flow. This is important. And if we have loading omission, like Dr. Jones says, then we have catastrophic failures in many other things. And the heart changes, and insulin resistance happens, and bones deplete. That's all happening in astronauts and people on bed rest for long term. The remediation of cutaneous properties through rehydration significantly improved tactile acuity. Such mechanical restoration of tactile ability has the potential to increase the quality of life in the elderly, but not just the elderly. Why do we have to wait until something is gone? If we lose Meissner corpuscles, if they're not as dense, how do we get them back? So we train the sense of touch. My favorite person in the whole wide world is Leonard Nielsen. Leonard Nielsen. This is his book, and this is him. And he has pictures of the ocean, of plants, insects, and the beginning of life. And he did this all on his own because he wanted to see inside the human body. So he made a special camera, because he's great at cameras ever since he was a little boy. He made a special camera that can go inside the body where there's a lot of liquid and actually take pictures that are good enough for us to see without being blurry. But here's one of his first masterpieces. Now, this was taken in the I'll get to it, 19 something. And the story is critical because he was on a polar bear expedition. No, he was on a whaling boat, and there was a polar bear, this little one, that was curious, swimming up to the ship. And as he tells it, you could tell the mom was kind of, no, don't go there. Don't go there, honey. This is not something that we can do. But this little one, not having the experience, wants to know what this big ship is. So, as the story goes, the mother polar bear came closer and she was shot. She was dragged up on the board of the boat, and being a little baby polar bear, this is the mother that had licked him and groomed him and done all the care and the soft touch, the slow touch, his whole life. And so as the picture is taken, you can see he needs comfort. He is scared. He doesn't know what's happening. He's on a ship. He's in a harness. It's never been on a harness before. And he's like, Mom, 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 I need you. And if that doesn't pull at your heartstrings and cause emotional change in your body through your vision and through the process of physical entrainment of hearing the story, I don't know what will. But what I can say is this picture went throughout the world, and it changed the whole way polar bears were hunted. Because people got emotional. They really did. Can you blame them? So Leonard Nielsen, he did that picture He was born in 1922. He died January 2017. He was taking pictures at an early age. He sold his first picture to a Swedish newspaper at the age of 16. When he turned 90, he donated all his photographic equipment to the Karolinska Institute, where he had worked since the 1970s. The picture of the polar bear, well, he took images of fetuses. So 1950s is somewhere where that polar bear picture was. Oh yeah, 1947. His picture from a polar bear hunting in Svalbard. I think that's in Norway. They were published in Life magazine, one of the most respected illustrated weeklies in the Western world, and caused so much debate. One of the pictures showed a polar bear clubbed with a rope around its neck, trying to hug its dead mother. So beautiful. We learned so much through our vision. And here is my daughter in 2005. Because she was conceived in 2005, and there was a tiny little egg, and this is a picture from Leonard Nilsson in his book Life. And yeah, pretty freaking incredible, isn't it? So this is the egg that was in me with all the DNA, the mitochondrial DNA, and here's the little instigator that started the whole process. And without this guy from my husband and her dad, she would not be here. So November 2005, this coupling happened. And now I got a fantastic, intelligent, a creative daughter who adapts and thinks on her own, and her whole body allostates on its own, and she knows when she needs to give herself a break. And that's what I'm doing right now. I'm giving a break, because we are done. Let's use the book because it's easier. So these are some of the images that Leonard Nilsson can come up with. Oh, let's move this screen. So that was my daughter with her little nutrient sac and her liquid around her, and that pressure is pushing in on her, so all her GRG neurons can sense that watery mess outside of her. And that's her fingers forming. Oh. And that's her little hand forming. And that is her all curled up in a tiny, tiny, tiny little place. And there's a lot of pressure. There's no way around it. That needs that kind of pressure in order to form and cause all sorts of things to happen. Pretty incredible, isn't it? I think I'm done. Yeah. It's beautiful. Absolutely beautiful. So, yeah, isn't he great? Fantastic what he accomplished all on his own, and probably without his camera, the endoscope that was used by Neil Feast to find out about the interstitium, and J.C. Gomberto to find out about the life of the inner side of the body, the fascia, strolling under the skin. I got it. So, thank you, Leonard Nilsson. Oh yeah, thank you. Thank you, thank you, thank you, Peter Sterling, for writing a great book. Thank you, Håkan, for sending this to me. I think I still have your note. Best regards. I learned so much. Thank you all. Robert, Andreas, got your work cut out for you. You're the scientist and engineer, and just bring the team together. So here's the information. Go have fun with it. I don't know, the chronic pain sort of thing and the neurodegeneration will be for another time, but I'm done. So we're done. Bye.
Thank you. So Gott.